So with that, I would like to first welcome Olivier Michelin. He is the head of the Precision Oncology Center in the Department of Oncology at the University Hospital in Lausanne. And he is the co-PI of one of our SPHN driver projects on the Swiss Personalized Oncology driver project. And I think looking forward to hear from Olivier how the project is actually dealing with all these challenges around data interoperability. So Olivier.
Thank you very much, Sabine. It's a great pleasure to have this opportunity to tell you more about what we're trying to develop in the oncology sector. I really highly appreciated the first two talks, and I hope this will give an example on how the analytics that has been described could be potentially used and helpful in oncology. So I think everyone is pretty much aware about the what is at stake in precision oncology, but I would like just to say that if you are able in a population like depicted here that has only a small fraction delivering a benefit, to identify the patient, i.e. to really find a biomarker that is able to pinpoint those patients that will deliver the long-term benefit, of course your treatment becomes an ideal scenario. You're only going to deliver it to the 20% with the long-term benefit. And the question is, how do you go beyond that? Well, you have to identify other patients that will benefit from other treatments upfront. So that's the grand goal of precision oncology, which of course will be ideal because patients will not lose time in treatment lines that are not effective. That might also reduce costs and of course rationalize the way we treat patients in an unprecedented way. Now, just a side note on what we're trying to achieve here. Usually knowledge in oncology and in every part of medical sciences is driven by prospective randomized phase 3 trials or smaller trials. To do so and to reduce statistical variability, one will usually come up with inclusion and exclusion criteria. And in this context, you can actually answer a question, is treatment A better than treatment B, with a high degree of confidence. And so no one questions the results of clinical trials. Here, in this case, experimental treatment is superior. The big problem we're facing in many places, and oncology being probably one of the forerunner here, is that we usually apply the conclusion of a trial to a much broader patient population. For example, brain mets were excluded, and then you treat patients, of course, who have brain mets because that's what you see walking through your doors. And so the question is, can we use real-world data to complement this? And that's exactly what we're trying to achieve with SPHN in the oncology field, is how can we use real-world data to complement the knowledge that we have from clinical trials. I think clearly what we need is to be able to get a lot of information for a given patient, clinical data, molecular data, whatever, omics. But per se, this is insufficient until you have a good reference where you can judge the alterations or the clinical scenario in which your patient lies. And it's only having these two types of information that you are able to build a personalized treatment program and AI that will be able to guide your decision. So that's the general context on where we were when we started this precision oncology, personalized oncology driver project of SPHN. And I would like to just give you, a few hints on where we stand. The first is when we started the program, we made a sort of a survey of what's the local infrastructure in each and every hospital. And of course, the heterogeneity was the only striking factor. We had various clinical databases, molecular databases, let alone imaging, omics. So this was really a big worry for us at the beginning. And how do you reconcile that with the way to be able to do data sharing, homogeneous queries in the whole country. And so we had to work hard at the beginning to define a minimal data set so that we can be fully interoperable. So most of the work that we have done in the first 2 years of this program lies in this first arrow here. How do we map our internal data into a commonly agreed minimal data set. And here, SPHN has been incredibly useful to define these data, to make them interoperable, and hopefully we are now at the stage where this is being filled, and we hope to move this into the whole country for query and for science very soon. Of course, what is very important is that you might want to be able to go beyond these structured minimal data sets. And of course, one of the road for the future is that we want to be able to query further into the different databases, for example, free text and different things. And I will show you an example on how we are trying to move that way. This program is actually the regroupment of 3 different driver projects, one from Mohamed Bentihas Ali for translational precision oncology, Mark Rubin for pathology in Bern, and myself for precision oncology. And this allowed us to gain the critical mass to move the program faster. I have to say it has been a truly exceptional collaboration with those 2 other PIs, and I think that the program goes very well. With this, we now have basically the 5 university hospitals on board. So we are exchanging, we hope to start exchanging data according to this minimal data set very soon. Actually has already started. And we have a 6th component into this network, which is the Swiss Cancer Research, SAKK, which has been phenomenal. I mean, we actually designed the program with SAKK directly with Roger von Moos. And I think this is a great addition because SAKK is connected to all the non-university and actually all the university hospitals for clinical trials. And so SAKK was really the natural partner for tapping into the non-university centers and being able to that way federate a data structure for the whole country. And as you will see, SAKK has been a fantastic player, and now there are initiatives actually originating from this collaboration that we hope will be transformative for the future. So what do we collect? We have here a very top level view of the main data. So code, ICD code, stage, treatment lines, whether the patient responded, yes or no, and then additional data, molecular data, imaging, and so on and so forth. These data has to be present for any oncologist anywhere to treat a patient. So our goal was really, instead of having data managers bringing this data into the minimal data set, is to change the way we capture clinical information in every single electronic patient record in every center, so that this data is already structured at the beginning. So when we hire the people for this program, we let each center free to hire someone from the IT, someone from the medical side, in order to complement what's already in place, so as to bring this information into the local data warehouses. And I have to say that this has worked beyond our expectations. So now we have a very good path in every university center, so that this data will become eventually fully automatized and be made available according to this minimal data set as clinical routine is proceeding. We have defined 5 different work packages. The first one for the continuous clinical data acquisition. WP2 is continuous molecular data acquisition. So this one now is completely handed to the program of Mark Rubin in pathology, which really deals with this with really great momentum. WP3 is how do we do that for regional hospitals. So this is now led by SAKK with very great momentum as well. WP4 is the Swiss Molecular Tumor Board, and WP5 is how do we add clinical research samples on top of this. I'd like to just show a very small excerpt of the different work packages. So this is a more detailed view of the items we collect for the clinical data set. Now we have 20 different items being collected. It's pretty standard. We add to this natural language processing, so this is being worked out as we speak. We try to mine pathology report, radiology reports, and I would like just to show you an example here with radiology report. I don't have time to go into the details, but just to show that using a very simple supervector machine, we can actually classify reports from the radiologist into progressively disease responding, complete response, yes, no, using pretty standard NLP techniques, as you can see here. So there is hope here that we can really use that to tap into the existing data and maybe also help on the prospective side, although for radiology, we hope to use a standard RESIST assisted PACS solution in the very near future. Now, Mark Rubin has been a fantastic player for the pathology side. You can see here the infrastructure that we have in place at CHUV under the umbrella of Sylvain Pradamon, who has been transformative here. You can see that we are using different data structure to at the end be able to do data mining for molecular data and clinical data at the same time. But at the Swiss level, Mark Rubin has pushed the installation of CBio portal from Memorial so that we can all have the same tool to view genomics data across the country. What is very important is we want this data structure to be able to be tuned for oncology. That means we need molecular data at the baseline when we start treating the patient, but also being able to look at how this data evolves as the disease is being reshaped by treatment. So sometimes we have those second biopsies, and so we need also a common data structure to look at time series and the way the molecular data are evolving with time. So this is also one of the important design parameters that we have in our head. And also being able to really tackle into a highly complex data set. It's just an example here of single cell RNA-seq, and you can see here a TC representation showing that you can from one single patient get a humongous amount of data. So we really need to be able to tap into this omics. But Mark Rubin has really paved the way on how to use genomics data for this work package and his own driver program. So I think this is going to be really a very nice opportunity to synchronize molecular data with clinical data. Now I'd like to just show you what we were able to build with SAKK, thanks to really an outstanding collaboration. The big difference between non-university and university hospital is the data structure. So usually hospitals in the university sector have data warehouses where it's less prevalent in the non-university, although this is changing fast, and that would be very nice actually to be able to use the same strategy for both, but this is only ongoing. So we have web-based solutions based on SECU trial, which was developed based on the SPHN minimal data set by SAKK, and very nice work that has been done here, which is now deployed. And the system that was put together by SAKK and SPHN is the so-called RWD, Real World Data Initiative, where basically the SAKK hospital, the non-university hospital, are inputting data according to the minimal data set, so as to feed different projects. And SPHN, basically the university hospital, comes in here to actually feed data as well, and both programs can exchange data. I mean, the governance is just in place, but in the very near future, we hope that this will be highly synergistic and a great opportunity for Switzerland, because that means the vast majority of the patient will be available and under the same description and the same nomenclature and the same norms in oncology. Last few points, Swiss Molecular Tumor Board. This is a nice initiative to try to help referring medical oncologists to take critical decisions. So we put together a Swiss-wide set of competencies, pathologists, bioinformaticians, geneticists, oncologists, many different experts, so as to bring an answer to a referring doctor on how to treat the patient given a set of omics. You can see that we have this activity here, for example, between Lausanne and Geneva. And just to give you an idea of the volumes, we have more than 350 patients per year just between these two cities. So it shows that precision oncology is really there. It's being used, and you can see that 70% of the cases are referred from outside of the university hospital. So that's really peripheral hospitals that are sending us patients for this type of analysis, which really shows that there is a need for this. You can see here an example of a success where we saw an amplification of PD-L1 in a patient with a rare coma, which is known to be completely resistant to immunotherapy. But this was the trigger to realize that this patient indeed needed to escape the immune system by this overexpression. So we treated the patient with PD-1, and the patient got into a complete remission, and he's still in complete remission despite being highly evolutive on chemotherapy. So these are the type of successes. Of course, not success every time, but still something quite useful at the moment. The question on how to transfer data and analyze data in a secured way, of course, comes around all the time. And we have here a very nice collaboration with Jean-Pierre Hubot and Jean-Louis Rezaro, who is on the call, who developed the MedCo infrastructure. So Jean-Louis will be much more capable of describing the internals, but just the idea that you can query different clinical centers in an encrypted way without leaving the data, yet being able to do general statistics like regression and all sorts of mathematics centrally, which is exactly what we need. So for example, if you are sitting on a tumor board and would like to know if treatment A is more efficient than treatment B, given a certain rules for patient selection, this might be the tool that can deliver and help us for clinical decisions. So this is a great addition to the project. There are also other tools being developed by DCC to do these cohort evaluations. So I think it's a great way for the future. One additional tool that we developed is a way to look at the impact of a mutation in 3D and using molecular modeling. This is a great collaboration with Professor Vincent Zoet. This website will be freely available in a few weeks, where you can see the mutation that you are interested in and the different neighbors. And so you can also assess whether your mutation will hamper the action of a given inhibitor, will activate your kinase, yes or no. So these are really key questions when you are sitting in a molecular tumor board. So as a conclusion, I think we are really witnessing a great era where probably moving the bar higher will require personalization of treatments. And we believe big data and the type of initiative that the SPHN and SAKK have spawned will really shape the future. And so with this, I'll stop this short presentation and thank all the university, hospitals, and structures that have contributed immensely to this, and most of all, SPHN for funding this whole program, which we hope will be very successful in the years to come. Thank you.